and researcher Rundy's with UC Davis and director of the Fragile X Research and Treatment Center of the Mind Institute. She knocks our audience off their feet every time she speaks. Oh. And we are so happy to have you back.
Oh,
I'm not going to list all your accomplishments and things, but
good.
You're going to talk about Fragile X, I take it, and a few other things.
Yeah, new treatments.
New treatments.
Yeah.
And then stay afterwards because we're going to get the afternoon speakers together to talk about what's next.
Oh.
Okay.
Okay. Wow, that's
Okay,
exciting.
That's your pointer.
Okay, my pointer and my advancer. Okay, so you guys all know about the Mind Institute, and we love it. So my disclosures are I'm carrying out a number of treatment trials. We just got funded to do the Zynerba trial about CBD, so I'll talk a little bit about that. Our Metformin study, which is a new targeted treatment for Fragile X and perhaps other forms of autism, is funded by the Azraeli Foundation. AFQ 056 is an mGlu-R5 antagonist, and we're funded by NIH, but AFQ 056 is made by Novartis, and we have a Fragile X Learn study with that, combined with using parent-implemented language intervention, or PILI, developed by Len Abadudo at the Mind. I'm also funded by Ovid to do a gaboxidol study. This is a new GABA agonist for individuals with Fragile X. And I've consulted with Zynerba to help them develop their protocol for the Fragile X treatment trials. So enough for disclosure. My overview is to really talk about the latest treatments for both Fragile X syndrome and for autism, and a future look at treatments for both disorders. Learning objectives, understand the relationship between Fragile X and autism spectrum disorders. Name a couple medications for new treatments for Fragile X, the same with autism, and to understand the difference in Fragile X between the pre-mutation and Fragile X syndrome. I follow a lot of individuals that have pre-mutation disorders like FXS, tremor ataxia, or Fragile X-associated tremor ataxia, and I have so many neurologists that see an older guy with a tremor ataxia syndrome associated with the premutation, and the neurologists say, "You don't look like you have Fragile X syndrome. You're a smart lawyer," and blah, blah, blah. And so a lot of people don't understand the difference between premutation disorders and full mutation disorders in Fragile X. And it's really Fragile X syndrome, those with a full mutation or more than 200 repeats, that I'll talk about related to these targeted treatments today. This is a new age of targeted treatments. It's been going on for the last decade, and we're very excited about that. Actually, things are moving into stem cell studies, being able to correct a genetic defect in stem cells and then insert it into patients. It's a very exciting new age, stem cell therapy, and gene manipulation therapies. So we're going to have a conference next year, probably a year and a half from now, at the Mind Institute about stem cell therapy for neurodevelopmental disorders. You hear a lot about stem cell therapy for neurodegenerative disorders, but not so much about neurodevelopmental. And there's so many bogus places out there that are injecting stem cells for autism, and it's just bull. And, you need to be able to sort out what's real, what's happening in the research for many genetic disorders. It's pretty exciting in the animal models, and I think in a few years it'll move more into humans. But before that takes place, we are really focused on targeted treatments that could reverse some of the neurobiological abnormalities related to different genetic disorders. And there's a lot of them going on in Fragile X syndrome. and I'll touch on each one of these, but also it's not just Fragile X syndrome, Angelman syndrome. Gaboxidol has some nice data for Angelman syndrome, and minocycline was somewhat helpful, but for Prader-Willi, you get diagnosed in the first year, you get put on growth hormone. I mean, this makes a real difference for that disorder. And tuberous sclerosis, of course, various mTOR inhibitors, including rapamycin, and even Rett syndrome. There's this paper coming out. I wrote an editorial for Neurology because the trofinetide treatments for Rett syndrome, this is the IGF-1 analog, there's a real nice study that's going to come out in Neurology, I think next month, about trofinetide treatment in Rett syndrome. And Anavex is a new treatment that looks really good in the Rett syndrome animal models. So these are new targeted treatments for many more disorders that you'll hear much more about. And some of these treatments are available, for instance, the Metformin that I'll talk about. But it's also important to think about the similarities across disorders. Like autism and Fragile X both have real significant GABA, glutamate imbalances, and many of disorders with the GABA deficits will do well, I think, with medications that really enhance GABA pathways, these inhibitory pathways, and I think that can help with behavior. I think just about every disorder that we look at has mitochondrial dysfunction associated with it. You're not having a mitochondrial mutation in the mitochondrial DNA, but the mitochondria don't work as well. It's very dysfunctional, and so people are doing more mitochondrial studies and thinking about medications that can improve mitochondrial abnormalities that impact the development of the child. Of course, all the aging syndromes, whether you talk about Alzheimer's or Parkinson's disease or whatever, all the neurodegenerative disorders, they all have mitochondrial abnormalities. And actually, everybody gets more mitochondrial dysfunction with aging. If you're older, you really feel weak in your extremities sometimes, and these are all related to mitochondrial changes that occur with aging. So oxidative stress, reactive oxygen species, and things that are dramatic antioxidants that can turn on the internal antioxidant system in cells, I think can really make a difference, and we'll maybe talk about that at the end. Environmental toxicity impacts many neurodegenerative or neurodevelopmental disorder, and also many neurodegenerative disorders. We study that in FACS-TAS and are very worried about general anesthesia and the isofluranes that cause more neuronal cell death, but we don't worry about it as much in neurodevelopmental syndromes, although I think it does have a big impact in autism and many other disorders. I'll talk a little bit about the Fragile X protein and how this can be a problem for many other disorders besides Fragile X syndrome. And, when you think about autism, and people have talked about the heterogeneity and, all of the comorbidities, and this has been such a great conference, and I'm sure you've heard a lot over the last couple of days, and many of the things that we use to treat autism is maybe focused on the mood instability or the ADHD or the anxiety, and we don't really have something that gets at the core deficits of autism with social and communication and these repetitive behavior problems. And of course, these are just a short list of all the different disorders associated with autism, and Fragile X is either the most common or the second most common cause of genetic disorders associated with autism. So whenever you see an autism patient, this is just to remind you about the workup. I'm sure you heard from Cindy Curry, not only about Fragile X DNA testing, but CGH array and battery. You can look out for ASD genes, and then so much more whole exome sequencing and whole genome sequencing. And the genetics people at UC Davis are just cranking on the whole genome sequencing and finding so many interesting mutations. We had an interesting adult not too long ago that turned out to have a mutation in a gene that causes iron deposition in the brain, and you could see it on his MRI, and it was a BPAP mutation. And this is the only cause of autism that I think benefits from chelation. And in fact, we put him on dysferapone, and he has gotten better as the iron has gotten out of his brain. But this is a specific mutation that causes iron deposition, and in adulthood, in his 20s, he started developing more Parkinsonian features and really deteriorating. And so he got better, but that was found through whole genome sequencing. So I just want to talk about how FMRP deficits can occur across many disorders, not only in those that have the full mutation of Fragile X or even the upper end of the pre-mutation, and the pre-mutation is 55 to 200 repeats, but actually studies of brains, particularly by Fatimi et al., have shown deficits of FMRP in the brains of individuals with autism, severe depression, and bipolar disorder, and most dramatically in schizophrenia. And in fact, the age of onset in schizophrenia and IQ in a population in Eastern Europe, in those papers there, correlate with the level of FMRP. So the lower the level of FMRP, and these are individuals without a Fragile X mutation, the lower the level of FMRP, the earlier the onset of schizophrenia, and the lower the IQ. Even tuberculosis neurons have been shown to have deficits of FMRP within the neuron. Hypoxia at birth lowers FMRP, and even early life seizures can significantly lower FMRP. So seizures actually pull FMRP away from the synapse, and FMRP is a protein that regulates translation of messages right at the synapse, and that relates to environmental stimuli. So it kind of turns on and regulates messages right at the synapse that are really important for synaptic plasticity. And when you have repetitive seizures, FMRP goes away from the synapse and towards the cell body. And maybe that's part of the reason why there can be cognitive deficits for individuals with recurrent seizures. But this just shows some of the brain studies in individuals with bipolar disorder there, depression and schizophrenia compared to control brains, and none of these have a mutation in the FMR1 gene or the Fragile X gene. And early life seizures pull these dots here are FMRP among the dendritic spines, and with early life seizures, FMRP, which is this green fluorescent, it goes away from the synapse and to the cell body. So when you dysregulate FMRP from the synapse, you're going to have some cognitive deficits. So FMRP not only controls translation, but it also is a messenger RNA carrier. It's one of these proteins that does just about everything. It even controls epigenetic regulation, and I'll show you that in a moment, or at least one of the proteins, BRD4, important for epigenetic changes in the DNA. It can enhance the release of neurotransmitters presynaptically much more rapidly so that the synapse gets flooded with the neurotransmitters so that individuals have, when there's an absence of FMRP, the synapse gets flooded and they can't detect subtle changes in the environment that are important for learning. It downregulates, when FMRP is absent, the GABA receptors are downregulated, and then it upregulates mGluR5 pathways. So it has a lot of effects, and I think we're just beginning to see some of the effects. So it's a big mother controller protein that when you mess with FMRP, you get a lot of effects in many different pathways. And we're learning more about it. It actually upregulates the insulin receptor, and I'll get more into that. But this is the effects on the BRD4 protein, which is really important for epigenetic regulation. And so FMRP usually inhibits translation, but sometimes it stimulates translation. And so when it's absent, there's an upregulation of proteins, including BRD4, which produces a lot more changes in transcription because of the epigenetic regulation, and then that in turn influences what happens at the synapse. So this interaction, epigenetic mechanisms with genetic and environmental influences is a very complex interaction that can cause some commonalities across disorders. And there's a lot of evidence for epigenetic dysregulation in individuals with autism, even in some of the twin studies, monozygotic twins, some of the methylation studies that we've done at UC Davis in Flora Tassoni's laboratory shows dramatic methylation changes in individuals with idiopathic autism, and whether that's related to environmental toxins or other factors in the environment, we don't know. I just want to show you just a partial list of some of the genes that are controlled by FMRP, and this circle are genes associated with autism spectrum disorders. These are all regulated by FMRP, and these are the genes associated with schizophrenia or mood disorders. So in about 10% of individuals with Fragile X, they can be on the psychotic spectrum. We've even seen psychosis in some individuals with the premutation, particularly those that smoke a lot of marijuana, which can push you towards the psychotic spectrum. So we're very interested in the interaction in those with a Fragile X mutation with psychosis, but you can also see psychosis. I've seen many aging ASD patients that have developed psychotic thinking over time too. That's not associated with smoking a lot of marijuana, but there may be other environmental toxins that can enhance that. Again, as I mentioned, many of the proteins or the messages that FMRP regulates, it binds to actually almost over 3,000 mRNA targets. Many of these genes, again, are associated with Fragile X or other disorders, Angelman, Prader-Willi, Rett syndrome. Actually, MECP2 controls FMRP expression, but FMRP controls the cannabinoid system also, so there's dysregulation in the cannabinoid system. I just want to show you this complex slide just to point out about the mTOR pathway. Here's tuberous sclerosis and this MEK-ERK pathway, and those with PTEN mutations can have big heads. This is neurofibromatosis, and these are all related to this mTOR pathway, and this is going to be important when I also talk about Metformin in a minute. So again, just to point out how common the Fragile X mutations are, basically about 1 in 200 females in this area. I just came back from Colombia in South America, and there are huge pockets of Fragile X in different villages there. In one village, 1 in 50 individuals has a full mutation, and more have the pre-mutation. And we found some other villages where there's just pockets of Fragile X, I think related to a founder effect. So, and many pre-mutation disorders there. And so the premutation disorders, it's the most common cause of early ovarian failure, most common genetic cause of early ovarian failure. FXS is the Fragile X-associated tremor ataxia syndrome, and it usually begins in the 50s or 60s, and affecting about 45 to 50% of the males and about 16% of the females. We've also identified the Fragile X-associated neuropsychiatric disorders, or FAXAND, because depression, anxiety, ADHD, obsessive compulsive disorder, chronic fatigue, chronic pain, and fibromyalgia are definitely associated with the premutation. So the premutation is common in the general population, whereas the full mutation of Fragile X is not, and the difference between the two is the level of messenger RNA. So premutation carriers, which are quite common in the general population, have dramatic elevation in the messenger RNA, as you can see there. And the full mutation, the whole gene gets methylated, so it shuts off message unless you're a girl and you have another normal X chromosome. But it's the lack of the message in the protein that causes Fragile X syndrome. And again, we have a subgroup of Fragile X that has a Prader-Willi-like phenotype with obesity and hyperphagia. And these are the individuals that I started treating first with Metformin, because it is a treatment for obesity and hyperphagia, and it helps them lose weight. But then we found some significant improvements in language and conversational speech, and that's why we're carrying out a control trial now. But hand flapping, hand biting, poor eye contact, even those that don't have a diagnosis of autism, and about 60% also meet criteria for autism, some of them that don't have autism and are very social can still have poor eye contact and hand flapping, and so some autistic features. So the GABA deficit causes a lack of habituation. So here we look at sweat response to repetitive stimuli, and usually whether it's a loud noise or a visual stimulus, you habituate in your sweat response to repetitive stimuli, but Fragile X don't, because they don't have a good GABA system. They don't have a good inhibitory system, and so they get activated with repetitive stimuli. So if you yell at them or try to spank them or whatever, you often will get into trouble with significant aggression. So you really have to go into a calming mode, as this guy is telling me non-verbally that I'm taking too many pictures of him. And so this is one of the reasons why, there's significant aggressive outbursts. They just get overwhelmed in overstimulating situations. And of course, they have macroorchidism, which begins at about 8 or 9 years of age, and these top lines represent about 180 patients we published many years ago, and these are the normals in terms of testicular volume here, but they usually have double-jointed thumbs, hyperextensibility, and this boy has a very subtle strabismus, but it led to amblyopia, and biting on your shirt and biting other things are quite common. So just getting into some of the targeted treatments, as we talked about, when FMRP is usually the inhibitor of this mGluR5 pathway, and when it's not there, there's upregulation of many proteins, including the proteins that internalize the AMPA receptors, and that's why you get long and immature and weak dendritic spines. So people thought, oh gosh, you just use an mGluR5 antagonist and you could cure this disorder, and that's what started the targeted treatment field. And we tried many mGluR5 antagonists, downregulating this MEK-ERK pathway and in turn maybe the mTOR pathway, but none of them really worked, particularly in adolescents and adults. We tried many other things. I'll touch on minocycline, ganaxolone. Allopregnanolone is a neurosteroid, arbaclofen, a GABA agonist. Even lithium will downregulate this pathway, but it doesn't cure Fragile X. So now it's thought that the earlier you start a targeted treatment, you will build a better brain on which you can apply learning. Okay, and so if you give an mGluR5 antagonist, in adulthood, which we have in many cases, sometimes they do better, but the overall study didn't show improvement. So now NIH through Neuronext funded an AFQ-056. It's an mGluR5 antagonist made by Novartis in a controlled trial. But on top of that, at the end of the study, they can go on open label. But on top of this controlled trial of AFQ-056, we added parent-implemented language intervention, also called PILI, which is done through Skype by a speech and language pathologist right into the patient's home. And basically what it's doing is it's teaching parents how to stimulate language at home, how to talk about the things that the kid's interested in, how to promote language, how to scaffold language. I mean, many of you know about Hannon techniques and other techniques. Well, it combines all of this. It's very effective alone for improving language in kids with Fragile X between the ages of 3 and 6. And we've been able to demonstrate that. And now we're analyzing the data. Well, actually, we're not finished with the study yet. We are not analyzing the data yet. We have until May to do the enrollment. It's at 14 different sites in the U.S., and hopefully we'll show some good results. We've had some kids, I don't know, it's a double-blind controlled trial, so I don't know what they're on, but many have improved. And so we're excited to look at the data and to analyze the data soon. It's led by Liz Berry-Kravitz, who's a really great neurologist at Rush University in Chicago. And then we're all helping her with this study, and Mind is one of the sites. So we did a minocycline study a few years ago now, and minocycline can downregulate matrix metalloproteinase 9, or MMP9, which is upregulated without the Fragile X protein around, and that interferes with synaptic plasticity. So if you bring it down to normal, it actually improves cognition and behavior in the Fragile X mice. And we did a controlled trial a few years ago, and we did show efficacy in blue on the CGI-I here. You can see that it was somewhat better. Of course, there was a big placebo effect, but we did get a significant effect. It wasn't dramatic, but we had a number of kids that did very well on minocycline. And most importantly, we developed an event-related potential. So this is an EEG hat that's put on that can look at habituation here, looking at habituation with placebo, and with the minocycline, and we brought back and improved habituation, also looking at some of the amplitudes which changed for the better, but habituation improved dramatically with minocycline. So we've been using that habituation paradigm in many of our outcome measures, along with some new expressive language sampling that Len Abadudo, who's the director at the Mind Institute, has developed. Now, in those with autism, there is in the brain a deficit of serotonin production, particularly in kids 5 or 6 years of age and younger, and compared to non-autistic children. This is a very old study by Chugani, but then there was some metabolomic studies that demonstrated, a paper by Boccaccio et al., found abnormal expression in the tryptophan metabolism pathway in individuals with autism. So here's tryptophan, and it gets metabolized up to serotonin in the brain, but all of these enzymes marked in blue are all dramatically downregulated compared in individuals with autism compared to controls. So that was another factor that suggested that the synthesis of serotonin in the brain is deficient in individuals with autism. Now, around that time, I also showed that in a retrospective study that those young children with Fragile X syndrome, when they were treated with sertraline or Zoloft, that their trajectory of language development was much better when I didn't treat them with sertraline. Now, the reason I was treating them is because anxiety kind of begins by the second year of life, and the anxiety can be very severe in individuals with Fragile X syndrome, and it seems to worsen social interactions. And here, when I treated them with sertraline, I saw an improvement in language. I didn't expect that. I was just treating the anxiety. And maybe if they were less anxious, that's why they talked more. But that's one of the reasons why we did a controlled trial. And this has been published now in Journal of Developmental and Behavioral Pediatrics, but it was a randomized controlled study. And what we saw was that on the Mullen, here's the reference here in Journal of Developmental and Behavioral Pediatrics. These are months improved on those on sertraline versus placebo, and we saw significance in many of the subtests on the Mullen. Not all. I expected to see the best effect in language, but about 60% of those in the study had autism, and a post-hoc analysis in looking at just those who had autism, that they were the ones that had a significant improvement in expressive language raw scores on this study. So I thought, wow, well, maybe we should be using sertraline in those individuals that have autism without Fragile X syndrome. And we got funded to do a low-dose sertraline study, 2 milligrams to 5 milligrams in kids 2 to 6 years of age with autism. The vast majority were idiopathic. We had a couple that had a known point mutation, but without Fragile X syndrome. And we used very similar outcome measures, including the Mullins. And it was a randomized controlled trial. Here's the consort diagram. And what we found is that looking at the CGI-I, this is sertraline in red, we had many that actually really did well on sertraline, and some looked like they even pulled out of their autism. But the overall significance was not there. So there's clearly just a subtype of those with idiopathic autism that seems to improve with sertraline. And we didn't see improvement in any of the Mullen measures. This is kind of broken up, but a lot of this was in the negative, that and those that were in the positive were not dramatic. So most of there weren't any significant findings on any of the Mullen subtests. That hasn't been published yet, but it is under revision now, also to Journal of Developmental and Behavioral Pediatrics. So we hope to finish that in the next few weeks. So why did sertraline work better in Fragile X than in idiopathic autism? Well, anxiety is a much more significant problem in individuals with Fragile X syndrome compared to idiopathic autism, even though anxiety can be a problem. But in Fragile X syndrome, it's a major driver of the severity of the ASD. Because anxiety leads to social isolation in individuals with Fragile X syndrome. And the level of the anxiety correlated with the level of the severity of autism in children with Fragile X syndrome. And then this very nice study came out just recently in Journal of Developmental Disorder, where they're looking at either pictures of faces. They're also looking at face processing, where you look on a face. And you remember that those with autism and also with Fragile X syndrome don't look at the eyes. They look at the nose and at the mouth. But there are social pictures and then abstract pictures here. And compared to typical controls, those with autism were less interested in the social pictures. Those with Fragile X were more interested in the social pictures. And in the face processing, there were differences. I can't see this so well, but the blue is Fragile X and the green is Autism. This is where they look on the face and how much time they spend looking at the face. But there are significant differences. And in Fragile X, there's a lot less fixation on the eyes. Supposedly that's overwhelming to the amygdala compared to those with autism. The interesting thing is that in Fragile X, those with more anxiety had less eye contact, but they were more interested in the social scenes and almost as if they were hyper vigilant to the social scenes. So that was very interesting, that those with Fragile X with more anxiety were even more interested in the social scenes, even though they might have had a diagnosis of autism. And that's very different for those with idiopathic autism. So let me get on to Metformin. It's a type 2 diabetes medication. It's known to help with overeating and obesity, and it's mainly used for elevated hemoglobin A1Cs in emerging and present type 2 diabetes. But in the Drosophila Fragile X model, they found that in Fragile X syndrome there was elevated insulin signaling, and that was brought down to normal with Metformin. And the PI3K, AKT, and mTOR pathway was all downregulated with Metformin, and it improved the circadian rhythm deficits and the memory problems in the Fragile X Drosophila model, and it also rescued the Fragile X phenotype in the Fragile X mouse model, the knockout mouse model. So at this time, when I was reading these papers in 2017, I thought, well, gosh, I should be using more Metformin in individuals with Fragile X syndrome, particularly those that are fat or have the Prader-Willi phenotype and hyperphagia. And indeed, they all lost weight. They improved their weight. But the parents told me, gosh, we're not so concerned about the weight. We really liked the fact that they could talk more, and I could finally carry out a conversation with my son. And so I thought, wow, maybe there are cognitive and language benefits. And so I read a lot more about Metformin. Basically, when you're on Metformin, it upregulates AMPK, and this inhibits this mTOR pathway. But it also has a lot of other effects, not only lowering the excess protein that's produced, but it has effects on the autophagy, effects on inflammation, and a lot of positive effects. It has protection because it lowers mTOR pathway. It protects you against breast cancer, cervical cancer, uterine cancer, ovarian cancer, and not just female cancers, but many male cancers. So I thought, wow, this is a really great drug. So I decided I was going to go on Metformin. I want to be protected against these cancers and live longer with better cognitive abilities to spend time playing with my grandchildren. And the main side effect is diarrhea. But I didn't have any diarrhea, and most patients don't have diarrhea, but you increase the dose slowly. We're doing a controlled trial, ages 6 to 25, that the FDA approved and gave us an IND. It's a randomized, double-blind crossover study, very easy study, only lasting 4 months, with just a visit at baseline, a visit at 2 months, and a visit at 4 months. So if you have any Fragile X patients, send them to us, because this and many other trials we have going, and we're using the best of the outcome measures we know, the NIH toolbox that David Hessel has organized to be more valid for those with intellectual disability. Behavioral measures, the event-related potentials, eye tracking with the Tobii eye tracker. We have molecular biomarkers, MMP9, and a lot of other downstream proteins on the mTOR pathway. And so I think I'm seeing good effects, but I'm blinded. We were funded by the Azrieli Foundation to also do it in 2 centers in Canada, Sébastien Jacquemont Center in Montreal, and Francois Boldec's center in Edmonton, in Canada. So they were just with us this week, so it's just a pleasure to work with the Canadian teams. So then I had a mom of this 2-year-old child who's a pediatrician at University of Utah, and she's a developmental and behavioral pediatrician. She says, after I presented some of the metformin data at Fragile X conference, she says, I want my 2-year-old son on metformin. And so I said, well, you know, it's not approved for 2-year-olds. And she said, I don't care. I want it in my son. And so we talked about using it clinically and talking about a safe dose, maybe just beginning at 50 milligrams once and then twice a day and sneaking up to a couple of 100 milligrams, you know, over the next year or so. And she found some amazing effects. You know, he was stacking 3 blocks, and then after a couple weeks on metformin, he was stacking 11 blocks, and she put that on her Facebook. And then the next day, I had a slew of emails from other mothers with 2-year-old kids. And actually, we've had a group of, actually, this is 9 kids here, and we're seeing some improvements in behavior. We looked at the developmental testing on the Mullen and compared it to what the developmental results of those with Fragile X syndrome not treated with metformin, and it looked like it boosted development about twice of what you would expect in Fragile X. So we think it has some good effects. It's not a cure-all. I just want to point out that usually there's IQ decline in Fragile X over the years, and it's because they don't keep up with abstract reasoning. So even though they don't lose skills, their IQ over time will plateau faster than the general population. We had this kid who had the Prader-Willi phenotype. We put him on metformin. He went back to the normal percentiles, but he was on metformin 2 years before he went into puberty. And then I saw him when he was Tanner stage 3, and his testicles were normal size. For those of you who know about testicle increase in Fragile X, it really increases to 2 to 3 times normal. So I thought, well, gosh, at least at the end of my career, I'm curing the macroorchidism in Fragile X. It's not a lot to be said for that, but I'd like to cure language. But then we've had a couple of adults here who have really boosted their IQ after a year on metformin. So we don't have all the results of the the controlled trial on metformin, but we're very hopeful it'll have some significant language benefits and maybe some cognitive benefits. So I just want to talk about some other new treatments for Fragile X and autism. The IGF-1 analog, which is trofinetide, has been beneficial in Fragile X in a previous study that we did, and it looks like it's very beneficial in Rett syndrome. We may be even starting an IGF-1 analog study in Rett syndrome at the Mind Institute in the near future. There's gaboxidol that I talked about. We're enrolling 13 to 22-year-olds with Fragile X. It's a controlled trial. Cannabidiol, or CBD, different from THC, or a tetrahydrocannabidiol, will likely be helpful from anecdotal information that I have from many families with autism and Fragile X syndrome for the anxiety and the tantrums and aggression that can occur, and they get it from the marijuana stores, but Zynerba synthesizes the CBD, so it's pure, it doesn't have any THC in it, and the blood is negative for THC, so the FDA really likes this. And it's an ointment that you rub on the shoulders and use it twice a day. So we're in the midst of that controlled trial now, and they'll be enrolling this spring, and hopefully we'll have the results in the very near future, we hope by next year. They have done studies in Australia, and I'll show you that data. These are open label studies in Australia. Let me just, wait a second. I don't need to talk about Epidiolex, but here's the Zynerba CBD studies in Australia, where each of these behavioral problems from behavioral checklists had significant benefit. But of course, an open label study is very different from a controlled trial. So I'm not sure what will be seen with the controlled trial, but I think it'll have benefit. I just want to mention about bumetanide. That's been going on in a study in Europe for autism. Bumetanide is a very old diuretic, and it is impacting the GABA system early on. It's a diuretic that can even be given to 1- and 2-year-olds. And I haven't had much experience. I've used it clinically. We don't have a controlled trial going now, but I think the people from France who started Lemonnier and Ben-Ari and others who started the bumetanide trial for autism in Europe want to get more studies going. They have a special liquid preparation of bumetanide, but it does come in 1 and 2 milligram tablets. And I use it on a 5-year-old, very severely autistic kid from actually Korea who was visiting me, and we're just desperate. And I got an email a month later, "This is an amazing drug, and my kid is talking," so maybe we should use it more. So think about bumetanide. You have to look for potassium deficiency that sometimes can happen with using a diuretic. So sometimes you have to supplement the potassium levels, but anecdotally, it seems to be quite good, and there's controlled data from the European studies that have been published, at least 2 papers on this, so it's definitely something we need to look at. Epidiolex, I'm not going to get into. You can get CBD gummies that are 25 milligrams per gummy on the internet, and when you go to the marijuana stores, though, they have all kinds of different solutions of CBD to THC, 20-1, and it's really hard to sort out how many milligrams are in some of the artisanal preparations, but I think many of these pure CBD preparations are coming out, and I'm not so sure you need to include THC. That seems to be the anecdotal information around that CBD won't work unless you have THC, but the data from Zenerba on the open label in Australia shows that that's not necessarily the case. So I'd be happy to talk to anybody. And oxytocin nasal spray, twice a day, and ASD, those with the lowest pre-treatment oxytocin levels benefited the most on the social reciprocity scale in autism. So I think there will be more use of the oxytocin nasal spray, but I'm not sure you can get it by prescription yet, and I haven't used it. So I also like yoga and mindfulness meditation. That really improves your GABA inhibition. This is a high-functioning Fragile X guy who does yoga every day, and he says that his life centers around it. And then also when we talk about targeted treatments, it's really important to think about educational programs. David Hessel has some data on CogMed intervention for individuals with Fragile X, Head Sprout programs for reading, AT devices, particularly for those that are nonverbal, iPad applications, and this PILI intervention, or parent implemented language intervention, or even Sally Rogers' Early Start Denver model, of course, is great. And there's a paper out a couple of months ago showing it's great in individuals with Fragile X syndrome too, not just in individuals with autism. So many great educational and behavioral problems. So my time is up and I got to stop, but I want to thank many of the people that we collaborate with, not only at the Mind Institute, but we have a lot of international collaborations in Indonesia, Philippines, where I'm going tonight, in Australia, Canada, and we do just a lot of international work, and thank you very much.